Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Or what do mitochondria do into those cells, and what is their function?
What happens in people when they have too much doxycycline or high doses? What happens to them?
Well, so they can get, muscle disruption or muscle dysfunction mostly, and other associated organs, because this antibiotic, what it does is eventually will disrupt the mitochondrial function itself. And therefore, the long use and high doses are not advisable.
What about from a histology perspective? Have you taken cells from these mice and counted the number of mitochondria on average and looked at their morphology under a microscope to see what's changed?
Yeah, so we have looked at in great detail, something we published. And so what we find that these mitochondria, one, they have abnormal cristae.
Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Keshav Singh, Ph.D. He's the Joy and Bill Harbert Endowed Chair and Professor of Genetics. We're going to talk about mitochondrial genetics and mitochondrial disease. He also has expertise in genetics, pathology, dermatology, and environmental health. He's the founding editor-in-chief of the Mitochondrial Journal as well, and the director of the Cancer Genetics Program. So very, very busy guy. Keshav, thanks for coming.
Delighted to be here, Richard.
If you would, tell me a bit about your current research. What are you working on?
So our main interest is obviously the mitochondria, and we're taking this mitochondrial genetics into different pathologies. So in the last couple of years, we discovered something which seemed to be pretty unique in the discovery we made. That was to do with mouse model we developed to identify the role of mitochondria in different organs. So far, what we know and knew about mitochondria was in isolated test tubes. We know it produces energy, it produces reactive oxygen species, but we made a mouse model where we can disrupt the mitochondrial function at will in any tissue or any organ or any cell type, and ask the question, what do mitochondria do into those cells, and what is their function?
When you say disrupt, what do you mean? Do you downregulate the metabolic function of the mitochondria, or what do you do specifically?
Yeah. So what we developed was a knockdown function of mitochondria where we took a dominant negative polymerase gamma. So in the mitochondria, you have one polymerase which drives the replication of mitochondrial DNA. And we looked at various different mutations in the last several years. And what we found was that one particular mutant functions as dominant negative. So we cloned that, we expressed that in the mice, in the whole animal. And what we found that these mice show first the disruption function. So therefore you have low enzymatic activity, the super complexes are downregulated. All of that eventually culminates into mitochondrial dysfunction.
Okay, so what what effects are you seeing when you disrupt mitochondrial function in mice? What are the immediate effects? What are slightly longer term effects?
So within few weeks, I would call it immediate, what we discovered was that mouse developed wrinkles and they lost their hair. And it was followed by other organ dysfunction as well. But that wrinkle formation and loss of hair was very striking. So we have been focusing on last several years, actually almost 2 to 3 years. What does it mean? Because one would think when we made these mice that these mice will die of cardiac problem or brain problem. They eventually do. And what also we can do is we can tweak the doses of the disruption. So based on the system we have where we can conditionally express the gene which disrupt the mitochondrial function, and depending on those, we can do less or more.
Okay. How do you do this disruption? Is this genetic engineering before the mice are born, or while they're alive? Is this like a gene therapy that does this?
So if you disrupt the gene which we are using, which is polymerase gamma, what happens that what's called embryonic lethal. So mice will die because it's an essential gene. So the way we went around asking the question about mitochondrial function in various different organs and tissues was do it conditionally. So when we take a dominant negative polymerase gamma mutant, it's only expressed when you feed the mice doxycycline, for example. And that way, we can turn on this gene and disrupt the mitochondrial function at any age of the mice. Obviously, if we do it too early in this embryonic lethal, the mice will die. So on average, we've been doing after 4 to 8 weeks, and we follow the study of those organs.
So you're saying, so 6 to 8 weeks, the mice are still considered young mice. Is that, do you inject them at that point, or what's this protocol look like?
So that's correct. So they are young mice. So what we do is we feed doxycycline in the chow. So whatever their food is, we add doxycycline to it. And then that doxycycline turns on the gene of interest, which will disrupt the mitochondrial function. Without doxycycline, that gene will not be expressed, and therefore you will not expect any disruption of mitochondrial function, only after when you feed the doxycycline.
I thought doxycycline in people was like a common antiparasitic or a drug that's been used for a while. Any of this analogous in people? Do they have the same effects?
That's right. So doxycycline is an antibiotic with, small doses. It has been used over, I would say, 10 to 15 years in animal studies that, it works fine. There's no issues in terms of the toxicity. If you take a higher dose, then it will affect like any, on humans or mice, same way.
Oh, so what happens in people when they have too much doxycycline or high doses? What happens to them?
Well, so they can get, muscle disruption or muscle dysfunction mostly and other associated organs because this antibiotic, what it does is eventually will disrupt the mitochondrial function itself. And therefore, the long use and high doses are not advisable.
Okay, so by disrupting the mitochondria, what are you figuring out in the mice? And again, can you look at data from humans and see analogs?
So what we do is when we disrupt it, we're looking at many different organs. The one we are focusing from the last 2 or 3 years is the skin and the hair. And because that's, there's a big market. Actually, I have a startup company called UA Biosciences. So that also fits in what we do in the laboratory into those research as well. So as you know, who wants wrinkle? As you age, what happens, your mitochondrial function declines. So this particular platform we have developed is keeping an eye on that area of research because as you age, your mitochondrial function decline, you have less of energy, and therefore also what you have is associated aging-related diseases. So the skin wrinkle also is part of the aging process, loss of hair, androgenic alopecia, you see when the androgen drops. So what we are focusing on is looking at the various different function of mitochondria in the skin. So one, obviously, we've shown the skin wrinkle. What we have also shown is the senile lentigens. So this is a fancy scientific name for hypo and hyperpigmentation of the skin. So as we age, you will see the skin in some patches you will have lighter skin color, other patches you are dark. And what we discovered that was also due to the mitochondrial dysfunction. So our mice clearly shows those hypo and hyperpigmentation. And then we're also looking at other effects of, in the skin. And what was striking, and that is what the basis of our company is, and that to do with the when we restore the mitochondrial function in the mice, so you can imagine when we feed doxycycline to the mice, it will turn on, it will disrupt mitochondrial function, and we will see the phenotype. But then when we take away the doxycycline from the food, from the chow, what we discovered was that it restored the skin to normal and the hair came back. So that was first time showing that a direct effect of mitochondria function involvement in skin wrinkle and loss of hair.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Is there an opposite compound that will boost the mitochondria, or does it not work like that?
No, so that's what we are looking at, and that's the basis of UR Biosciences. So there's a drug discovery program, we have artificial intelligence platform we are using, so that's the goal. And it will not only help with the restoration of the hair and mitigation of the skin wrinkle, it will also help in terms of other diseases as well. There's a whole field of mitochondrial diseases which are supposedly until recently were called rare diseases, but the combined effect of those diseases, like 1 in 2,000 children born have mitochondrial disease, and the primary cause of disease is mitochondrial dysfunction. And unfortunately, there is not much for those children or adults who suffer from the diseases. We'll be trying to help those patients as well if we discover how to restore the mitochondrial function in that area as well.
What about from a histology perspective? Have you taken cells from these mice and counted the number of mitochondria on average and looked at their morphology under a microscope to see what's changed?
Yeah, so we have looked at, in great detail, something we published. And so what we find that these mitochondria, one, they have abnormal cristae. And what happens due to the mitochondrial dysfunction, the immune cells get infiltrate in the skin, epidermis, and they actually lead to the inflammation of the skin. Also, we have looked at mice shows other genes which are involved in aging, for example, clotho, which is downregulated. So the mitochondria themselves, the number doesn't change because we are not interfering with the number per se. We are interfering with the function of mitochondria by depleting the mitochondrial DNA content.
Okay, so the number of mitochondria appears to stay the same. Does anyone know the biogenesis rate of mitochondria and how many are present in mouse or human cells at any one time on average? I'm sure it depends on tissue type, but what are some averages?
Well, so is anybody's guess. I don't think we have the tool to exactly find the number of mitochondria. So say, for example, in human oocyte, it can be anywhere 200,000 mitochondria. And then you combine that with the mitochondrial DNA in those mitochondrion. So if you have 200,000 mitochondria and then say 10 times more mitochondrial DNA in each one of those, so it becomes actually a population genetics within the cell. But I would not be guessing how many mitochondria, but we do know it differs from tissue to tissue. For example, when we knock down the function, as I said, we were expecting that, the heart and lung and brain would be affected because they require a lot more energy and they require a lot more mitochondria. But turns out it has major effect on the skin. And the skin cells don't have that many mitochondria as the heart and lung. So what it tells us that those whatever mitochondria number there is in the skin is extremely important and regulated. And if you mess up with those mitochondria in the skin, you get a strong phenotype.
Well, maybe because of the low number, there's fewer mitochondria to take up the slack, and therefore you have a pronounced effect.
That's right.
Is there a way to, I don't know, with the expression of some compound or the metabolization of another compound, are you able to approximate the number of mitochondria in a cell without being invasive?
No, not really. I mean, you can look at within the cell. The other problem in counting the number is mitochondria undergo fission and fusion. So at any given time, when this fission and fusion is going on, that number is changing within millisecond. So how do you actually count a number, and what you count will be given at that time, at that moment. So we need a better technology to count the number, and one can develop a microscopy, or videography where you do it over the long period of time in a certain cell type and try to do it in vivo and in different organs and different cell type. But that we are not there yet.
What causes fission? What causes fusion? Has anyone established that?
Yeah, so that depends on the metabolic demand of the cell. So, if the mitochondria are injured, that metabolic demand will change, and they will basically change their fission and fusion capacity.
Well, under what circumstances is there more fusion, and under what circumstances is there more fission? And biogenesis. I mean, does biogenesis come from the act of fission, or is it a third process?
So one can imagine that the biogenesis can come through the act of fission, but it can also be by nuclear control of increasing the number of mitochondria by other means. Fission can, basically create more mitochondria, but the process also involves getting rid of that damaged or dysfunctional mitochondria. So in the fission, you can chop that off, and that can go through the mitophagy and get cleaned up. That's one way of maintaining the quality control. But there are mechanisms to restore mitochondrial function in the cell, and we actually published several years ago where we talked about epigenetic control of nuclear DNA by mitochondria. So when we disrupted the mitochondrial function, there were a set of genes which are turned on, and actually they methylated the nuclear genome. And we have identified a couple of microRNA and other things which are. So I gave an idea of what we call mitochondria checkpoint. Later it became the metabolic checkpoint. Others used it and broadened it. And the idea there is that if there is mitochondrial dysfunction, the cells have to make a cell-fate decision. In other words, mitochondria, given the involvement of apoptosis, the cell have to decide if there is really big damage and they cannot repair the mitochondrial function, they have to trigger a pathway to basically go under apoptosis and cell death. But the checkpoint will try to restore that mitochondrial function. And the one our contribution was looking at methylation of the nuclear genes and DNA.
So, I don't know, again, that's a whole other catalog, I guess, of up or down regulation based on epigenetic effects of mitochondria. I'm sure there's probably volumes and volumes to be written on that. Anything that jumps out at you? What is a significant contributor to the epigenetic changes that happen in mitochondria specifically?
So you mean the mitochondria driving the epigenetics, or epigenetic driving the mitochondria?
Well, what kind of effects? I mean, what do various drugs or what do various effects have specifically on, methylation or demethylation of mitochondria and their up or down regulation of their activities?
The methylation of the mitochondrial DNA is controversial. There are some groups that have shown that their mitochondrial DNA is methylated, but some have not been able to reproduce it. But there is likely to be in certain circumstances of physiological state that mitochondrial DNA is methylated. They have shown, actually a group in Virginia showed, I cannot remember the name at the moment, is that there is a DNMT, which is a methyltransferase isoform which localizes to mitochondria. And the idea was that particular DNMT, the one which may methylate the mitochondrial DNA. But other way around, where we showed that disruption in mitochondrial function lead to large number, we utilized restriction landmark genomic scanning to map the entire genome in the nucleus. And we found hundreds of those genes were methylated. And that's where the idea came from, the mitochondrial So I would think that, it's going both directions.
The effects of doxycycline, have they been quantified on exactly how they affect the mitochondria? Do you know down to the genes and to the method of action on what's happening with them, or is it still somewhat mysterious?
So it is somewhat known, and that is the way doxycycline does, that it interferes with the ribosomal biosynthesis. So these mitochondria have their own ribosome, and over the time, if you use for too long, I use the doses which are toxic, that will lead to the inhibition of the ribosomes in the mitochondria, and therefore they cannot drive the protein synthesis in the mitochondria. So that's how it will interfere. There's enough known about it.
Okay. What about the fission or fusion cycle? Maybe maybe it predisposes mitochondria to fuse. Could that be determined, or does it push more for fission? Like that balance of fission, fusion, biogenesis. How is that affected, do you think, by doxycycline or other compounds?
I don't know that. I have to look at it, but I would think that dysfunctional mitochondria will certainly affect fission and fusion. So I'm not sure which direction, or which will be more or which will be less.
I mean, experimentally, how could one determine that? Do you think that's important, or is it just like, not very important?
Well,
how does anyone determine these dynamics?
Well, it's not really important per se, but now I'm curious to see if anything in the literature. But what we know is the mode of action of doxycycline. So the direct target is the ribosomal biosynthesis. So when that is being inhibited, therefore your mitochondria are not going to work properly if you have high doses or for a long time. So whether the cells are adapting through fission, fusion, methylation, and all that is secondary to the effect what doxycycline is doing.
I don't know. What headway do you think you're going to make over the next couple of years? Are you close on the tail of answering any of these questions you're working on, or what do you think is likely to be figured out by you?
Well, so we are on to a couple of things using the platform we have developed. One, obviously, we want to follow up with UR Biosciences, is to look at skin wrinkle and hair loss and skin aging. Obviously, we are interested in pigmentation because also one can imagine the freckle. When you go out in the sun, you get freckles, and there's nothing out there to understand how freckles form and what you can do about it. Most of those sun creams, they don't protect you as much as one would think. So that's one area, but we have also embarked on ovarian aging. So these mice, what we have developed, show premature ovarian aging. And there's a interest from my laboratory and globally to extend the menopause, to understand how ovarian aging occurs, because ovaries age faster than the woman, chronologically speaking. So we have got clear idea that how disruption of mitochondrial function is doing that. And obviously, I'm interested in another reproductive organ that's to do with the prostate. So that's the work in progress. And these are all part of the, aging process. So when women, around 50, they have menopause, obviously the ovaries have aged. For men, it's the same thing as the prostate. So those three things, I think, is fascinating to me, and also it will lead to some translational research which can help people.
Okay, very good. Well, Keshav, where can people find out more about your work? Where can they go?
So one can look into keshavsingh.org. So that's my name without the middle initial, .org. Or, in fact, if you just type mitochondria.org, I took this domain name actually in 1999.
Oh, wow.
And actually, I used to have .com as well, but I gave it away, so a friend of mine has it. Mitochondria.org is me.
Wow. That's really cool. That was nice of you. That's a great gift,
Yeah.
Huh. Excellent. Well, very, very good. Keshav, great to talk to you. And thank you for what you do and for what you're working on.
Well, same here. Thank you.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at FindingGeniusPodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.